All right, so chapter 34 really should probably be 2 or more chapters, but our book lumps circulation and gas exchange together, and we'll talk about how those things tie in together coming up. So when we think about things that need to trade places, resources and waste need to be able to trade places. So resources that cells require, like nutrients and oxygen, have to enter the cytoplasm by crossing the cell membrane. Unicellular organisms are pretty simple. They can exchange things directly with their environment, but multicellular organisms, such as ourselves, rely on specialized systems for transport and exchange. Exchange organs could be like the gills, which exchange oxygen from the water into the blood vessels and carbon dioxide from the blood into the water. And our book lumped these two topics together because really transport and gas exchange are pretty functionally related in most animals and are linked by the circulatory systems of many animals. So small molecules like oxygen and carbon dioxide have to move between cells and their immediate surroundings by diffusion, but diffusion really doesn't work well over long distances. Diffusion time is proportional to the square of the distances traveled. So diffusion is really only going to be practical over small distances. So we have to have a circulatory system to link the exchange surfaces with cells so that that material can be exchanged at cells as well as the outside of the body. In some animals, the body places many or all of its cells in direct contact with the environment. So each cell can exchange materials directly with the environment around it. Some animals have a gastrovascular cavity. We've talked about this before. Remember the cnidarians or the jellyfish with one opening that functions as both the mouth and the anus. We talked about it in kind of the digestion section, but in this case, we're going to talk about it more with distribution of substances. So gastrovascular cavity serves both digestive and circulatory type activities. And then circulatory systems are found in animals with more complex body plans, and we can have either an open or a closed circulatory system. The circulatory system has a circulatory fluid and vessels. A muscular pump, which like we have a heart, is going to power circulation by using metabolic energy. An open circulatory system is found in insects and other arthropods and some mollusks, and open circulatory systems don't have blood. Instead, they have hemolymph, and hemolymph is also the interstitial fluid. Remember, interstitial, inter is between, so it's the between cell fluid. So in this case, we're saying if you don't have a closed circulatory system, like all of your circulatory vessels don't loop together, then your extracellular fluid is going to be the same as the stuff that's in that circulatory system. So then if we go to closed circulatory systems, annelids, which remember that's your segmented worm, cephalopods, which are like squid or octopi, and all vertebrates have a closed circulatory system. And we have blood because it's confined to our vessels, and our blood is different from our interstitial fluid. So our blood and the fluid between our cells is different. We have one heart. Some organisms have multiple hearts to pump blood through the vessels, and then chemical exchange is going to occur between the blood and the interstitial fluid, and between the interstitial fluid and the body cells. So let's review. The first circulatory system up there in our segmented worm is an open or closed system. It's closed. Let's go to the one to the right on top. So that would be like an insect there. So that's going to be an open circulatory system. If we look at like the insect on the bottom left, still an insect, and they're showing you like the tubes are not all connected there, so that's another open circulatory system. And then our totally anatomically correct picture of the fish there is trying to show us a closed circulatory system. So humans and other vertebrates have closed circulatory systems called the cardiovascular system, and the main types of blood vessels are arteries, veins, and capillaries, and blood is going to be one way in these vessels. So if we look at the blood vessels, arteries are going to be conducting blood away from the heart. Arteries have thick muscular walls and they're going to be under higher pressure. And then an arteriole is basically a small diameter artery, and it can constrict or dilate to control blood distribution. Then we have capillaries, which are our smallest cells, and they have thin walls. They're for gas, nutrient, and waste exchange. And if you take anatomy or physiology, you're probably going to get into more detail because there's multiple kinds of capillaries, but for now we're just going with capillary. And then you've got venules, which are small, thin extensions of the capillaries, and then you have veins, which conduct blood back to the heart, and they're less muscular than arteries and contain valves. So if we look at the picture here on the left-hand side, we've got an artery, and up at the top of the picture, they've got a cross-section of the artery, and you can see it's got thicker muscular walls, and then in between the artery and arteriole and the venule, there's the capillary bed, and then you've got the venule, and you can see that the venule has a valve to prevent backflow of blood in the veins. And if you look at that cross-section up at the top, you can see that the vein has a thinner wall as well. So then if we look at vertebrate hearts are going to have 2 or more chambers. Blood is going to enter through the atria, and then it's going to be pumped out through the ventricle. Then we'll talk about different kinds of hearts coming up here. And then septa, or a septum, is going to separate the atria and the ventricles. So with closed circulatory systems, you can either have a single circulation or a double circulation, and there's a couple variations on double circulation. So fish have a single circulation where they have a single atrium and a single ventricle and kind of one big loop that the blood takes. With double circulation, you have 2 loops that the blood is going to take, and we'll talk about each of these coming up here in a moment. So with a single circulation, this is going to be found in things like bony fishes and rays and sharks, and these guys have a 2-chambered heart with 1 atrium and 1 ventricle. The blood takes kind of one big loop, or a single circulation, and the blood actually has to pass through 2 capillary beds before it returns to the heart. So the blood leaves the ventricle and goes to the gills. At the gills, it's going to pick up oxygen, and then the blood goes through another capillary bed in the body where it's going to drop that oxygen off, and then it goes back to the heart and is picked up in the atrium. I said before there were kind of 2 variations on double circulation. One of them is double circulation with intermittent breathers. So an intermittent breather is something like a frog that can do some gas exchange through its body surfaces rather than just its lungs. So these critters are going to have a 3-chambered heart with 2 atria and 1 ventricle. And the ventricle is going to pump blood into a forked artery that splits the ventricle's output into the pulmocutaneous circuit and the systemic circuit. So the pulmocutaneous circuit is the lungs and the capillaries in the skin. That's for gas exchange. And then the systemic circuit is to the body, which is also for gas exchange, but we're doing gas exchange kind of in an opposite way. So in the pulmocutaneous circuit, it's picking up oxygen, dropping off CO2, and in the systemic capillaries, just like in our systemic capillaries, the cells are going to be picking up the oxygen from the blood, and the blood is going to be picking up carbon dioxide from the cells. And in these guys, the blood can actually be nearly stopped from going to the lungs. Obviously it has to have some, but the blood flow to the lungs can be vastly reduced. Turtles, snakes, and lizards have a 3-chambered heart with 2 atria and 1 ventricle. And again, that circulatory system allows kind of control of the relative amounts of blood flowing to the lungs and the body. But in alligators and caimans and other crocodilians, they have a septum that divides the ventricle, basically providing 4 chambers. And then you've got a connection to the atrial valves that can temporarily prevent blood flow to the lungs as needed. If you think about these things, life histories and what they need to do, like think about how long an alligator or a crocodilian needs to be able to stay underwater, and that is a good adaptation for them. So with double circulation, this is probably what you've thought of before when you thought about circulation in the heart before. So in this case, oxygen poor and oxygen rich blood is going to be pumped separately from the right and left sides of the heart. The right side of the heart delivers oxygen poor blood to the capillary beds of the gas exchange tissues, and then the left side of the heart propels the oxygen rich blood to the capillary beds in organs and tissues. And then the oxygen poor blood is now going to return to the heart and complete the systemic circuit. So double circulation can maintain higher blood pressure in the organs than a single circulation would. So in mammals, the right ventricle is going to pump blood to the lungs by way of the pulmonary arteries, and that's going to pick up oxygen and unload carbon dioxide in the lungs. And then blood from the lungs is going to enter the heart at the left atrium by way of the pulmonary veins, and it's going to be pumped through the aorta to the body tissues by the left ventricle. And then the aorta is going to provide blood to the heart through the coronary arteries, and diffusion of oxygen and carbon dioxide is going to take place in the tissues, and the blood is going to return to the heart through the superior vena cava and inferior vena cava, and it's going to enter in the right atrium. So I think this picture is an okay picture, but it's a little bit complicated. So on the next slide, we've got a simplified version for you. So here's our simplified diagram of mammalian circulation. So I'm going to start with a box. So we've got the box there, It's labeled RA and LA and RV and LV. RA stands for right atrium, LA stands for left atrium, RV, right ventricle, LV, left ventricle. So I would encourage you to draw it on a piece of paper, but you can kind of look at it and get the idea here. So you might notice that I drew the box on the left ventricle thicker than the rest of them. If you remember from our heart dissection, the left ventricle has a thicker myocardium, so a thicker muscular layer, because it's delivering blood to the body. So blood goes from the left ventricle to the body by way of an artery. That artery is going to be the aorta. And obviously this is simplified. You have way more blood vessels than what we're going to show you, but this is like the major blood vessels involved. So then from the body, you do gas exchange in the tissues and, nutrient exchange in the tissues. And then it returns back to the heart. Oops, there we go, back to the heart at the right atrium. And it returns to the right atrium at the superior and inferior vena cava. Then from the right atrium, it goes to the right ventricle. Then from the right ventricle, goes to the lungs, and it takes the pulmonary arteries to the lungs. And then from the lungs, we pick up oxygen, drop off carbon dioxide, and then go back to the heart. And we go back to, sorry, my buttons are hard here to push apparently. We go back to the left atrium, and you ride the pulmonary veins to get there. So how do we prevent backflow though? We've got valves. The one between the left atrium and the left ventricle is the left AV valve. The one on the right side between the right atrium and the right ventricle is the right AV valve. But how do we prevent blood from the pulmonary arteries and the aorta from getting back to the heart? Valves again. Notice the moon shape. So these are the semilunar valves. The one that goes to the pulmonary artery is the pulmonary semilunar valve, and the one that goes to the aorta is the aortic semilunar valve. So that's our kind of simplified structure of the mammalian heart. So if we look again to kind of review, we've got single circulation, double circulation in amphibians with the pulmocutaneous circuit, and the double circulation that we're going to find in mammals. So the fish has a 2-chambered heart with 1 atrium and 1 ventricle. The amphibian has a 3-chambered heart with 2 atria and 1 ventricle. And then the mammal, like ourselves, has a 4-chambered heart with 2 atria and 2 ventricles. So we have a little bit of a mammal bias in our class. So let's look closer at the mammalian heart. So as the heart contracts, it's going to pump blood, and as the heart relaxes, it's going to have the chambers fill with blood. And one complete sequence of pumping and filling is called the cardiac cycle. The contraction phase is called systole, and the relaxation phase is diastole. So if we look at this diagram, on the left-hand side, there's the number 1, and it says atrial and ventricular diastole. So that means both of those are relaxed, and you can see they're showing with the arrows that the blood is filling the heart. If we go to the next one, you've got atrial systole and ventricular diastole. So the atria are contracting and the ventricles are relaxed at that point. And then the next one, you've got ventricular systole and atrial diastole. So the ventricles are contracting and the atria are relaxing. So the atria are filling with more blood, and the ventricles have contracted to pump blood out. So that's kind of the complete event. And they've got timing there on the slide. I don't care that you know the timing, but it's good to know the order and how this works. So let's review. Which system contains hemolymph? Is it a gastrovascular system, an open circulatory system, or a closed circulatory system? If you said B, great job, right? So an open circulatory system has hemolymph. What's hemolymph again? Remember, hemolymph is the same thing as the interstitial fluid. If you have an open circulatory system, the fluid between your cells is not any different from what your, quote, blood would be, so it's all hemolymph. In a closed circulatory system, we have blood and interstitial fluid, and they are different from each other. Number 3, how many chambers does a mammalian heart have? We've got a 4-chambered heart. We've got 2 atria and 2 ventricles, so 2 and 2 is 4 still. Number 4, which kind of closed circulatory system has a pulmocutaneous circulation? Is it single, double intermittent, or double? You might wonder, what does pulmocutaneous mean? So remember, that is when the blood can flow either to the lungs or the skin for gas exchange. So that's going to be in that double intermittent breather, like the frog. Number 5, when is blood pressure highest in the systemic circuit? When the ventricles contract or when the ventricles relax? It's going to be when the ventricles are going to contract because they're pushing the blood out of the heart, and that's going to increase that blood pressure. So if we keep looking at this mammalian heart, cardiac output is determined by the heart rate, which is your beats per minute, and the stroke volume, which is the amount of blood pumped by ventricle in one contraction. But if you think about it, that makes sense, right? Like, how much blood can the heart pump? Well, how often does it beat, and how much blood can it pump out in one beat, right? So that's not too terrible. We've already talked about valves in our beautiful diagram a few slides back. So valves are important because they prevent the backflow of blood in the heart. And we have the AV valves, which separate each atrium and ventricle, and the semilunar valves, which prevent blood from flowing back from the aorta and the pulmonary artery. And then when you hear a heartbeat, the sound of the heartbeat is actually caused by the blood hitting the AV valves and then the semilunar valves. So it has kind of a characteristics like lub-dub noise. And if it doesn't sound like that, if you have kind of a swooshing type noise, that can be a heart murmur, which is caused by backflow of blood through a defective valve. So the heart needs to beat an a rhythmic beat, and cardiac muscle cells are actually autorhythmic, meaning they don't need a signal from the nervous system to contract. Basically, they can leak ions, and then that causes them to contract. But they also do have input from the nervous system. So the cyanoatrial node, or the SA node, is sometimes called the pacemaker, and that sets the rate and timing at which the other cardiac muscle cells are going to contract. And the SA node produces an electrical impulse that's spread rapidly through the heart. And if you've ever seen like an electrocardiogram, that's what that's tracking there. And then impulses from the SA node go to the AV node, or the atrioventricular node, and then the AV node passes the message on to the Purkinje fibers, and the Purkinje fibers are actually what makes the ventricles contract. So if we look in this slide here, it's got an image of electrocardiogram, so it's showing you the SA node, and then the SA node triggers the AV node, and then the AV node triggers the bundle branches, and then the Purkinje fibers, and that actually causes the ventricles to contract. Obviously, there's other cues that can alter your heart rate. Hormones and temperature and physiological stress can all alter the heart rate as well. So then blood flow velocity, you can think of velocity as like the rate of flow. So blood vessel diameter is going to influence blood flow. A more open blood vessel is going to have more blood flow through it than a more narrow blood vessel. So blood flow is actually going to be slowest in the capillary beds. The capillary beds have very small diameters, and they have a huge cross-sectional area because there's so many capillaries. But this slow flow of blood is actually good in the capillaries because we need to do gas exchange, and we need to exchange waste and nutrients in the capillaries as well. If you look at the graph on the right hand side, the top one is showing you the area, and you can see that there's a more cross-sectional area of capillaries, which that makes sense. There's a whole lot of capillaries compared to really large arteries or very large veins. And then the next one is showing you velocity in centimeters per second. So the arteries have a higher velocity than the capillaries. The veins have a slightly higher velocity than capillaries, but they're in between arteries and capillaries. And then pressure, they're showing you in the arteries your systolic and diastolic pressure because you've got the direct effects of the heart beating there. And then they're showing you pressure goes down as you go from large diameter arteries all the way to the capillaries, and then even to the veins and vena cava there. So blood flow and pressure and resistance are all kind of related. So blood pressure is the force exerted by the blood on the walls of the blood vessels, and blood flows from areas of higher pressure to areas of lower pressure. Resistance is the tendency of blood vessels to slow down the flow of blood. This is based on vessel radius or diameter, vessel length, and also blood viscosity. I think a lot of people don't know what viscosity is. So viscosity is resistance to flow. So something that would be viscous would be like molasses on a cold day, right? It resists flow. It will stay in the bottle, whereas water is less viscous than molasses. So if blood is more viscous, it's going to have a higher resistance. But really, nobody has molasses in their blood, or at least hopefully nobody has molasses blood. So for most organisms, radius is the most important factor, or like diameter, right? right? The radius is just half the diameter. So how wide the vessel is. So vessels can vasodilate or vasoconstrict. If they vasodilate, they get larger because the smooth muscles relax, and so that increases the radius. And I don't care that you know this, but nitric oxide is a major inducer of vasodilation. And then vasoconstriction is when the blood vessel constricts and becomes smaller because you've got contraction of smooth muscles. This decreases the radius, and this is going to reduce flow. And again, I don't care that you know this, but peptide endothelin is an important inducer of vasoconstriction. All right, so we got the blood out of the heart, into the arteries, to the capillaries. We did some exchange, and now we have to get the blood back to the heart. So venous return is getting that blood back to the heart. So we already said the pressure is lower here, but we do have valves that prevent backflow of blood. Gravity is a pretty important consideration with blood return. If we're standing up, our blood is going to be drawn towards our feet instead of towards our heart, but we have these valves in our veins to prevent that backflow. We also have contraction of smooth muscles in the walls of veins and venules, and contraction of nearby skeletal muscles that helps blood be kind of squeezed through those veins and get back to the heart. If you've ever seen like a winter program choir thing, or soldiers standing at attention for long periods of time, sometimes somebody falls off the back of the risers and it's awful. And yeah, that happens because they lock their knees, right? Or maybe you never saw anyone do that, but maybe your choir teacher was like, don't lock your knees, but hold still and stand up straight. And you're like, how do I do that? But either way, if you don't have any skeletal muscle contraction in your legs and you lock your knees, that can impair blood flow to your head. So you're not getting enough blood to your brain. Your brain really likes to have blood in it, as it turns out. And your brain is like, oh, this is a problem, but I know how we can fix this. We can just lay right back down, and then we're not fighting gravity anymore. So that's part of why fainting happens. An animal with a long neck, like a giraffe, has to have a very high systolic pressure to pump blood against gravity all the way up to their brain there. So then capillaries are really critical for exchange of substances, and the blood flow in the capillary can vary. Blood is really typically only flowing through like 5 to 10% of the body's capillaries at a particular time. However, capillaries in major organs are usually pretty full. You can alter blood flow into capillary beds by either vasoconstriction or vasodilation of the arteriole that supplies that capillary bed. There's also pre-capillary sphincters. A sphincter is just a ring of smooth muscle, so that it can like open or close the entrance to that capillary bed, and so that can regulate the passage of blood to that particular capillary bed. So blood is going to enter the capillary under pressure, and pressure is going to force some of the fluid out of the blood. But typically red blood cells and large proteins are going to stay in because they're going to be confined by the capillaries themselves. Most of the fluid that leaves the capillary is going to be recaptured at the venule end of the capillary. The pressure is going to decrease along that capillary bed, and the proteins in there are going to create an osmotic force that actually draws the fluid back into the blood in the capillaries, right? So there's a high solute concentration in the capillary, lower solute concentration outside the capillary because of the difference in that protein concentration. So the fluid will come back into the blood at that venule end of the capillary. However, not all of it comes back. Some of it will have to be collected by the lymphatic system, and we'll talk more about the lymphatic system coming up. So here's a very simplified picture. We've got an arteriole on the left, and there's one tube in the middle that's supposed to be our capillary, and then a venule on the right hand side. So blood entering the capillaries are under higher pressure because you've got that pressure from the arteriole, and so that's going to push out water and solutes. However, proteins and large cells should stay in the capillary. And then on the other end, the lower pressure end, water and solutes are going to come back into the capillary because there's a high concentration of protein, so water and solutes can enter into the capillary there. But anything that doesn't enter back there is going to be picked up by the lymphatic vessels, which are those green tubes in this picture. And eventually the lymphatics are going to return the lymph to the blood. So the lymphatic system returns fluid to the circulatory system. While the fluid is in the lymphatic system, it's called lymph, and lymph is very similar to the interstitial fluid. But think about that. Where did the lymph come from, right? The lymph came from the interstitial fluid. And remember, interstitial is just the between cells fluid. So lymph is basically extracellular fluid that has been picked up and put into the lymphatic vessels. And eventually the lymph will drain into veins in the neck and be returned to circulation. So the lymph vessels have valves to prevent backflow, and you've probably heard of lymph nodes before. These are organs that help filter lymph and play an important role in the body's immune system. And we'll talk more about the lymphatic system in the immune chapter as well. So here's just kind of a quick overview of some lymphoid organs. On the left side there with the thymus and the appendix and Peyer's patches in the small intestine and lymph nodes and the spleen and lymphatic vessels and the adenoids and the tonsils. And then on the top right, they're showing you the lymphatic vessels with their valves, and they describe them as like blind-ended tubes. So they have an end to the tubes. They're not all connected in a loop like the circulatory system is. And then at the bottom on the right hand side, you've got a lymph node. If you look at the lymph node, this picture did a good job showing you that you have more pathways in than out of the lymph node. So that gives the fluid time to be kind of dealt with. If you imagine filling up a pool with like 4 hoses and you only had 1 hose out of the pool, the water would fill up in the pool and the filter would be able to work. So that brings us to the components of blood. Blood is actually a fluid connective tissue. Those of you who've taken anatomy know that there's a whole bunch of different kinds of connective tissues, but blood is a type of connective tissue, even though people tend to think like bone, tendon when they think connective tissue. Blood contains nutrients and proteins and gases and blood cells as well. So plasma is the liquid portion of the blood that contains water and solutes. Plasma plays a role in buffering and transport of the cells and also water balance. Then you've got leukocytes, which are your white blood cells. They help defend against infection and disease. You've got erythrocytes, which are your red blood cells, and they're important for oxygen transport. Ours use hemoglobin. They don't all use hemoglobin, but that's what ours use. And then you've got thrombocytes, which is the $5 word for platelet, and platelets are important for helping form blood clots. So this slide just shows you kind of your stem cells that create your blood cells in your bone marrow, and then the different kinds of blood cells. For our class though, I'm not going to ask you to identify the difference between like a neutrophil or a basophil or any of that, but I do want you to know erythrocytes are red blood cells, leukocytes are white blood cells, and then platelets. So again, erythrocytes are red blood cells. They're the most numerous blood cell you're going to have. They typically contain hemoglobin. Sometimes they don't contain hemoglobin. We'll talk about other respiratory pigments coming up here, but if they had hemoglobin, it would help them transport oxygen. And there's a whole feedback mechanism that's sensitive to oxygen levels that controls your erythrocyte production. So if you've heard of cyclist blood doping, it's because they either increase their amount of erythrocytes by stimulating their production, or they increase their amount of erythrocytes by like taking their own blood. That would be a really beneficial thing for them since they're endurance athletes, but generally I would not encourage you to do that. Then leukocytes are your white blood cells. They engulf bacteria and debris, and they're important for fighting foreign substances, and they can be found both inside and outside of the circulatory system. They can actually leave the circulatory system to go hunt down invaders. So then platelets are fragments of cells and function in blood clotting, and basically they form a solid clot and a liquid blood. And this takes a cascade of complex reactions that converts an inactive form of fibrinogen to fibrin, and then that forms the framework of the clot. And then a blood clot that's formed within a vessel is actually not a good deal. That's called a thrombus, and that can block blood flow. And it depends on where it is, how big of a problem it is, but generally speaking, not a good thing. And on the picture on the right, you can see red blood cells. They're those kind of they're called a biconcave disc, but that red pancake looking thing is your red blood cell, and then you can see the threads of fibrin there. So we really oversimplify things a lot in this class, but basically clotting factors are made from platelets, damaged cells, and your blood plasma itself, and these set off an enzymatic cascade where prothrombin is converted to thrombin, and thrombin keeps that thing going, that cascade going, and then the thrombin converts fibrinogen to fibrin, and you get a fibrin clot formed. So we talked before about positive feedback cycles and the platelet plug formation. So the platelet plug itself is not the total clot. You also have to have this fibrin clot forming to kind of hold everything in place. So let's review again. How would the following factors alter resistance? Would they increase or decrease resistance? So would vasodilation increase or decrease resistance? So remember, what is vasodilation? Vasodilation is increasing the radius or the diameter of the blood vessel. So if the blood vessel gets bigger, what's that going to do to resistance? That should decrease resistance. If we increase the number of white blood cells or red blood cells, which increase blood viscosity, what would that do? Would that increase or decrease resistance? So remember, viscosity is resistance to flow. So if we had more blood cells that increase viscosity, that would increase resistance. So number 7 then, in a closed circulatory system, blood is going to be pumped away from the heart, but what helps the blood return to the heart? So how do we get blood in the veins back to the heart? There's valves that prevent backflow of blood. You've got contraction of the skeletal muscles around those veins that kind of squeeze the blood back to the heart. Maybe if you've taken anatomy, you've heard of like the thoracic pumping. So like when you breathe, right, it changes the diameter of your chest cavity, and that can help as well. So if you wrote something like that on the test, I would take it, but we didn't. I guess we just covered it now, but it's not in our book, but it's a thing that happens. So those would be 3 important factors there. And then 8, students often say arteries carry oxygen-rich blood, or arteries carry oxygenated blood. Is that a correct statement? So not entirely, right? The arteries in the pulmonary circuit, what do they carry? So the arteries in the pulmonary circuit, where do arteries come from? Arteries are going away from the heart. So in the pulmonary circuit, that's the blood going to the lungs. So is the blood going to the lungs, is it rich in oxygen? It is not. So arteries in the pulmonary circuit carry oxygen poor blood. So be careful on that. Rather than saying arteries carry oxygen rich blood, you should think arteries move blood away from the heart. Or I always think of like an artery wearing a cape, like arteries away. Hopefully that helps. So that brings us to physical properties of gases. So now our book is kind of switched to the gas exchange section here. So if we look at air is composed of oxygen, nitrogen, and then like carbon dioxide and other gases. It's mostly nitrogen, but as far as we're concerned, we care about the oxygen content of the air. So we usually just kind of ignore nitrogen because it's not really part of this respiratory process. So gas exchange is the movement of carbon dioxide and oxygen between the air and the blood, and then between the blood and the cells. So we've got like a super simplified picture on the right hand side here. They've got the external environment where like really that's inside of our lungs. It's weird to think of inside of our lungs as the external environment, but that's the air that it's basically dealing with. So we're exchanging oxygen from the external environment into the cells of the respiratory organ, and then carbon dioxide is leaving, and then the oxygen is picked up by the blood, and then the oxygen is picked up by the body's tissues, and then the body's tissues are going to do cell respiration, right? They're going to make CO2, they're going to use that oxygen, so then the CO2 is dropped off into the blood, and then dropped off into the respiratory organ system cells, and then into the external environment. So to understand this in more detail, you have to understand partial pressure of gases. In physics, you'll probably talk about this more. We're going to kind of summarize it here, kind of tell you some half-truths, but true enough to get us by. So in a gas, all the gases in like a mixture of gases like air is going to exert some pressure, and the pressure that one gas exerts is called its partial pressure. So atmospheric pressure is the sum of the partial pressures of all the gases in air, and air is 21% oxygen, so the partial pressure of oxygen, or the PO2, is 0.21 times 760 millimeters of mercury, which is atmospheric pressure, and so you get 160 millimeters of mercury. Really, we can think of a gas's partial pressure as kind of like the concentration of that gas in the mixture of gases. So partial pressure also applies to gases dissolved in liquid like water. So when water is exposed to air, an equilibrium is reached in which the partial pressure of each gas is the same in the water and the air. I'm sure you've all either drank a soda or a pop. Right? Where are we from? I don't know which one you guys say, but you know what I'm talking about. You've had a soda or a pop, you've cracked it open, right? You've drank it, and then if you put it to the side, like what's going to happen to it? Right? It's going to go flat. Why did it go flat? Because the carbon dioxide that they had shoved into the can has come out into the air. So there's equilibrium there. So we know from before, like diffusion is driven by concentration gradients. So with gases, when we're dealing with gases, diffusion is just driven by partial pressure gradients. You already know this. It just kind of overcomplicates it to some degree with partial pressure because people get hung up on that. But just think concentration when you hear partial pressure and you will be okay. So then the rate of oxygen diffusion into blood is lower at higher elevations because atmospheric pressure is lower at higher elevations, even though the amount of oxygen in the air is the same. So atmospheric pressure is the pressure exerted on animals by the atmosphere. It's not just on animals, right? But we're talking biology, so that's really all we're caring about in this particular case. But that would not be the same definition for atmospheric pressure in a physics class, right? But you measure pressure in millimeters of mercury or kilopascals, which are kPa. Millimeters of mercury is mmHg, right? Millimeters of mercury. And atmospheric pressure decreases at higher elevations. At sea level, atmospheric pressure is about 760 millimeters of mercury, but think about Mount Everest, right? I would not want to go up Mount Everest. But the reason people bring oxygen tanks up to Mount Everest is because the pressure is so much lower, they have a hard time doing gas exchange there. So the percentage of the gases remains more or less the same regardless of altitude, but the lower atmospheric pressure results in lower partial pressures, which makes it harder to do gas exchange. So we have different gas exchange mechanisms depending on what kind of critter we are. So gas exchange can happen across the surface of an organism's body. It can happen across gills. It can happen through tracheae or through lungs like we have. And ventilation is the process of bringing the respiratory medium, which is either going to be water or air, into contact with those gas exchange surfaces. So gas exchange is going to take place by diffusion. So there's like no active transport of oxygen. It is diffusion for oxygen and carbon dioxide transport. And all respiratory organs are going to have some things in common. They're all going to have a moist surface, so the gases can dissolve and then diffuse, and then they have a high surface area. For like the more surface area you have, the more gas exchange you can do, and then they tend to have a thin, delicate structure because again that's going to increase the surface area. And then they have, if they have blood, they're going to have extensive blood flow to those organs. And not everybody has blood, right? Some organisms have hemolymph. So keep that in mind. So then if we think about the difference between breathing in water and breathing in air, well, obviously we would have a big problem breathing in water because we've not evolved to do that. But broadly speaking, aquatic animals and air-breathing animals have different challenges. So aquatic animals have less available oxygen than air-breathing animals. And when the temperature changes in the water, the oxygen availability also is going to change. If you fish, you know that you're going to find different fish in warm water and cool water, and that warm water is just really not as hopping because warm water doesn't have as much oxygen availability there. Aquatic animals also have problems moving water over their respiratory membranes to some degree because it takes more effort than moving air. Whenever I say that, I always think of my mom does water aerobics. It takes more effort to move dense water than it does just air. And I'll tell you, I've gone to water aerobics with my mom before, and like it is hard to move water, and you know that too. And then the water also is going to remove heat from the gill surface and can create osmotic movement. That should probably be 2 bullets because that's 2 different things, right? You've got temperature regulation problems and then osmotic movement. You're like, oh, osmotic movement, no big deal. No, it's a huge deal, right? Drawing water either out of your cells if it's salt water or having water rush into your cells and causing your cells to lyse, that's a big deal. So those are challenges aquatic animals have had to deal with. Terrestrial animals like ourselves, so land-dwelling critters, our biggest problem is desiccation, which you're like, oh, we only have one thing on our list. Yeah, we only have one thing on our list because desiccation is a big deal. So like you don't want your respiratory membrane to dry out. Imagine your lungs drying out and becoming crispy. That'd be awful. So that is our challenge as terrestrial animals. So some organisms can do gas exchange just across their body surfaces. If you only have a few cell layers, you can just use diffusion and you don't have to have a specialized transport system. But some large animals, like amphibians, have skin that allows them to do gas exchange. The chart on the right-hand side here has a bunch of different organisms and how much oxygen and carbon dioxide they can exchange across their body surfaces. So the lungless salamander, as you would assume, since it doesn't have lungs, it is going to do all of its gas exchange across its body surfaces. Bullfrog does more carbon dioxide exchange. And then if you go all the way down to, let's look at the southern musk turtle. The southern musk turtle can do gas exchange across some of its body surfaces as well. And if we go down to like the brown trout, it's not doing much gas exchange across its body surfaces because what's a trout use? Right? Gills. You got it. And then a human, we don't do much gas exchange across our surfaces either. So what is the biggest difficulty terrestrial organisms have with their respiratory system? Our biggest challenge, like doing gas exchange on land, is drying out our respiratory surfaces. So got to keep the lungs moist. What do aquatic organisms have for problems? So aquatic organisms have to move dense water. They have to be able to regulate their temperature. They've got to be able to regulate the amount of water moving in or out of their cells depending on the environment that they're living in. And then also when the temperature of the water changes, the oxygen that's dissolved in the water changes as well. So if it's a higher temperature, there's less dissolved oxygen. So external gills then, they vary pretty widely in appearance, but they all have a large surface area and they're all external, so they're projections. They could exist in one spot or be scattered across the body depending on what kind of critter you are. However, these things wave around in the water and they are totally unprotected and subject to damage. They can attract predators to chomp on them. They look delicious, right? If you're a predator, they do. And then also it requires energy to wave those things back and forth. So internal gills are probably what you think of when you think of gills. Fish gills are covered by a bony operculum, and different fish ventilate their gills different ways. Some of them do buccal pumping. Basically, they open their mouth, that draws water in, and then they open their operculum, and that draws water through. Another way is ram ventilation, which basically if you swim with your mouth open, right, that's going to push water into your mouth and then move it past your gills. So these are flow-through systems. Water moves unidirectionally. It goes from the mouth, over the gills, and out the fish. Here's the old countercurrent exchange system. They have a countercurrent exchange of water and blood flow to maximize oxygen diffusion in the blood. We've got a picture of that on the next slide here. So if you look, the first picture with the fish is showing you the direction of water flow, and then the picture below that, they're showing you a gill arch with lamella and secondary lamella, and then you've got a blood vessel going out to the tip of that and a blood vessel going back in. So the efferent blood vessel is what is returning to the fish's body, and you can see that's red. They're showing you that it's red to show you that it's highly oxygenated and it's picking up what little oxygen it can add at the kind of fresh end of the water. And then on the other side you have the afferent with a blood vessel, and that one is getting the slightly more oxygen poor water, but it's still able to pick up oxygen, and it kind of gets the oxygen picked up before it goes to the efferent blood vessel. So that's a countercurrent exchange, just like we had with the the bird foot in the cold water with temperature and the loop of Henle in the kidneys. So then for another gas exchange system, so the tracheal system in insects consists of a network of air tubes that branch throughout the body. So they have spiracles on the body surface that lead to tracheae that branch into tracheoles. And the really cool thing about this is that they terminate near every body cell. So insects have a tube that connects to another tube that connects to another tube that connects to the cells. And that transports oxygen and carbon dioxide without having to participate with the circulatory system. So then we've got lungs, which are probably what you think of. Lungs are enfoldings of the body surface, usually divided into numerous pockets. I think I got that from a textbook somewhere. It's fun to think of them as pockets, right? And then our circulatory system is going to transport gases between the lungs and the rest of the body. Depending on what kind of critter you are, your lungs and ventilation is going to vary. Lungs can be filled either using positive or negative pressure, and lungs can be ventilated either using tidal ventilation or flow-through ventilation. So positive pressure ventilation is found in amphibians and a few species of reptile. And what happens here is they open their mouth, and that opening of their mouth expands that cavity and creates a pressure gradient, so air moves from the outside of their body into their mouth, and then they are going to close their mouth, and that is going to force air into their lungs as if inflating a balloon. With a negative pressure ventilation, this is found in mammals and birds, and the big picture on how this works is that the chest cavity is going to expand, and so that's going to increase the volume of the lung, so it's going to have a lower pressure, so then that's going to draw air into the lungs. And then to get the air out of the lungs, the chest cavity will decrease in size and the air will be forced out. So mammals such as ourselves use tidal ventilation, and our lungs never completely deflate, or at least they really shouldn't. They are held open because they're adhered to the chest wall, and that is why a newborn mammal's first breath is a very hard breath to take and a very big deal. Birds have a flow-through ventilation system where they have air sacs that function kind of like a bellows that keeps the air flowing through the lungs, and air passes through the lungs of the bird in one direction only. And it takes 2 cycles of inhalation and exhalation to get the air through the whole system there. And really, our book doesn't talk much about birds at all. I just think it's interesting, so I added it. So don't worry about birds. So here's a picture of how we breathe, basically. So a big one is the diaphragm. It's the muscle that separates your abdominal cavity and your thoracic cavity. And if you remember doing like the rat dissection, you probably remember seeing the diaphragm. So when we inhale, the diaphragm is going to lower during inhalation. So it's going to expand our chest cavity. Also, your external intercostal muscles, which are the muscles between your ribs, are going to contract, and that's going to increase the chest volume as well. So when you inhale, your chest volume is larger, your muscles have contracted, so air will be flowing into your lungs because the pressure there is lower than outside. And then when you relax those muscles, air is going to flow out because the volume of the thoracic cavity will be smaller, and so that'll push the air out of the lungs. So let's review. Match the type of ventilation. Is it flow through, tidal, or buccal pumping? And is it a positive or negative pressure system? So the first series of pictures on the left hand side is a human. So do we have flow through, tidal, or buccal pumping? We have tidal ventilation. And then is it a positive or negative pressure system? It is negative pressure because we're increasing our thoracic cavity, and that is pushing, or that's not pushing, that is like sucking the air into our lungs. Let's look at the frog. So the frog opens its mouth and kind of like gulps the air into its lungs. So that is going to be buccal pumping, and that is a positive pressure system. The bird, I said don't worry about the bird for the test, but it's an interesting one. So the bird is a flow-through system, and it is negative pressure. So let's take a closer look at the mammalian respiratory system. So this should be relatively straightforward, and on the test these make good like put these in order type questions. So if we start with the nose and the mouth, the job of the nose and the mouth is to warm and humidify the air and also clean it. If you've ever like sanded something without wearing a mask, you will know that or been in like a dusty area, you will know that the nose is important for helping to clean air. Then you've got the pharynx, which is the fancy word for your throat. Then you have the larynx, which holds your vocal cords, and you have the glottis, which is the opening to the trachea, and that's protected by the epiglottis. Then you have the trachea, which has rings of cartilage, and also cilia and mucus to trap particles and sweep that mucus up. The cilia do the sweeping. This cilia sweep the mucus up to protect the surface of the lungs. So then if we get to the lungs are surrounded by a pleural sac, and the trachea is going to branch into two bronchi, and then it goes to bronchioles, which really, there's a whole lot more branching than this, but our book is heavily simplifying. So you've got bronchioles that have circular muscle that can dilate or constrict the passages, and then eventually you get to the alveoli, which are the little sacs for gas exchange, and these are 1 cell thick, so they're thin, which is good for diffusion. And then if we go back to that pleural sac is 2 layers of thin moist tissue with a fluid between the layers. That fluid acts as a lubricant, and that helps to prevent friction. So right, your lungs are moving a lot, you don't want friction there, it can cause tissue damage. And if you don't believe me, if you rub your hands together long enough, right, they'll get hot, and if you keep rubbing them, right, you'll get tissue damage eventually. So that is your lungs. So the next part here is about coordinating circulation and gas exchange, and I feel like going over this actually overcomplicates it, and sometimes you already know it and get more stuck. So stay with me here. But the blood that arrives in the lungs has a low partial pressure of oxygen and a high partial pressure of carbon dioxide relative to the air and the alveoli of the lungs. In the alveoli of the lungs, the oxygen is going to diffuse into the blood, and carbon dioxide is going to diffuse into the air, right? So the blood has dropped off CO2 and picked up oxygen, which I think you knew that before this slide. And then in the tissues, the partial pressure gradients are going to favor diffusion of oxygen into the interstitial fluids and carbon dioxide into the blood, which again, you knew that before this slide. You know that at the tissues need to pick up oxygen because they need to use the oxygen, and the tissues produce CO2, so they drop that off into the blood. And there's specialized carrier proteins that play a role in this process. Okay, so this next one I think is a little bit tricky, but stick with me here. So if we go number 1 on the right hand side, it's talking about air that you're inhaling from outside of your body. So that has a high partial pressure of oxygen and a low partial pressure of carbon dioxide. And then if we look at the air in the alveoli, since we don't empty our lungs entirely, right, our partial pressure of oxygen is a little bit lower than atmosphere, and our partial pressure of carbon dioxide is pretty high, a lot higher than atmosphere. If we look at the alveolar capillaries, those capillaries are going to be able to pick up oxygen and give CO2 off to the alveoli. If we look at the pulmonary veins and systemic arteries, their partial pressure of oxygen and partial pressure of CO2 is about the same. If we get to the systemic capillaries, those are going to drop off oxygen at the body tissues and pick up CO2 because the body tissues have a low partial pressure of oxygen and a high partial pressure of CO2. And then if we go back to the pulmonary arteries and systemic veins. Their partial pressure of oxygen, partial pressure of carbon dioxide are similar to that of body tissues. And then if we look at exhaled air, we're getting rid of CO2, so we have a higher partial pressure of CO2 and we have a lower partial pressure of oxygen than inhaled air would have. So respiratory pigments are going to be used to help transport oxygen and in some cases carbon dioxide in the blood, or hemolymph if you don't have blood. Remember again, hemolymph is the word for what we would call blood in organisms with an open circulatory system. And there's a variety of respiratory pigments, but they usually have a metal bound to a protein. So the two that we're going to talk about are hemoglobin and hemocyanin. Hemoglobin is what we have. Oxygen is going to bind reversibly, and then we can move it around other parts of the body. Hemocyanin is not going to be in a red blood cell. It's directly in the hemolymph, and it's blue on the PowerPoint because the central atom is copper instead of iron like hemoglobin, and that makes oxygen-rich blood blue. This is found in mollusks and crustaceans. But we really, again, are mammal-centric here, so we're going to talk about hemoglobin. So when oxygen binds to one subunit of hemoglobin, it changes the shape slightly, and it increases the affinity for oxygen for the rest of the subunit. So when oxygen binds, it's easier for more oxygen to bind. But then when one subunit releases oxygen, the others release their bound oxygen more easily as well. When we're doing a lot of cellular respiration, we produce a lot of carbon dioxide, and carbon dioxide, remember, is at equilibrium with carbonic acid and bicarbonate ion. So more CO2 increases the carbonic acid concentration, so it will lower blood pH or make blood pH more acidic, and that decreases the affinity of hemoglobin for oxygen. But this is a good thing, right? Because when you're making a boat ton of CO2, it means you're doing a lot of cell respiration, so you need more oxygen. So instead of having that oxygen tied up in hemoglobin, it is available for the cells to take in. Hemoglobin also helps prevent harmful changes in blood pH and plays a little bit of a role in carbon dioxide transport. So if we look at the partial pressure of tissues during exercise and tissues at rest in your lungs, you can see that it's very easy for the oxygen to be unloaded to tissues during exercise. And if we look at the pH and hemoglobin dissociation, hemoglobin is going to retain less oxygen at a lower pH. So hemoglobin lets go of oxygen easier when there's a high carbon dioxide concentration. So to transport carbon dioxide, most carbon dioxide is going to diffuse into the blood, and it's transported in the blood plasma as like a bicarbonate ion or attached to hemoglobin. So let's review. Place the structures in order, starting with where air enters. So I'll give you a minute to look at that. All right, so where does air enter? We would start at the nose and mouth, and then we would go from the nose and mouth to the pharynx, and then to the trachea, and then to the bronchi, and then to the bronchioles, and to the alveoli. What did I forget to include? The larynx. So really we'd go nose, mouth, pharynx, larynx, trachea, and so on. Number 14, compare the image of the normal lung to the lung with emphysema. How would this affect an individual? So normal lung on the left, diseased lung on the right. They pointed out broken alveoli for you. So how would that affect an individual? Well, you'd have less surface area for gas exchange. And then, how would you feel if you didn't do as much gas exchange? You'd be tired, you'd be short of breath, right? You'd be tired because you aren't getting enough oxygen to make enough ATP. So that's kind of what I was looking for with that one. Looky there, you made it through your lecture. I know it's a long one. So we talked about circulatory systems, we talked about numbers of chambers in hearts, talked about the cardiac cycle, blood flow through the heart, and then we took like a side note to like blood vessels and their functions and the components of blood. Started on the lymphatic system, but we'll get more to that later. And then we talked about gas exchange and all of that. So if you have any questions, definitely feel free to reach out to me. I'm absolutely happy to help. I will talk to you on the next chapter.